So today, what I'm going to go over is essentially protein folding. Okay, so we're going to talk about some of the little details and some of the mechanisms that happen when we talk about how proteins form a functional structure. In particular, like we know enzymes, structures are very important. Okay, so we're going to talk about that. We'll get into a little bit of quaternary structure. In particular, quaternary structure is essentially many monomeric protein come together and form a dimer or trimer, tetramer, that kind of stuff, right? That's quaternary structure, okay? So that's the scope of today's class. All right. Right off the bat, I'm going to tell you what denaturation is, okay? So we know that proteins, okay, when it's made, okay, when it's being translated and when it's being produced inside of your cell, they are folded, okay? And they're folded into a particular structure that is able to do whatever it does. Okay, now, when you take the protein and you throw it into some harsh conditions, then the protein is going to lose its three-dimensional structure, okay? And this process is called denaturation, okay? Naturation is like natural, what it's supposed to be. Denaturation meaning you get rid of its natural structure and you sort of break the protein. You don't fragment the protein, but you destroy their tertiary and their secondary structures, okay? So, denaturing condition, okay, so we're going to talk about what are the denaturing conditions. They include several things you can do to a protein to denature it. Of course, the easiest thing would be increasing the temperature. Okay, so we're going to talk a lot about what temperature has to do with protein folding and proteins' free energy. Okay, so obviously increasing the temperature of your protein, it's going to make it sort of move very fast, and then it's going to break whatever internal hydrogen bond or hydrophobic effect, whatever interaction that holds the protein together. It's going to break that apart, okay? Or when pH becomes extremely acidic or alkaline, okay? So obviously, if you throw a lot of protons or a lot of hydroxide anion into the solution, that's going to interact with your protein significantly, okay? So that will hydrolyze some of the bonds, okay? Reduce the amount of hydrogen bonding you can have intramolecularly. So that will denature your protein. Or a lot of times you can use some kind of organic solvent, for example, SDS, right? When we talked about how to look at proteins biomass, we saw that we can use SDS. You guys remember what SDS is, right? 12 carbon with a sulfate at the end, right? That 12 carbon sort of can go into your protein, expand it out, so it'll break your protein apart and make it into linear, okay? So that's sort of a detergent type of thing, organic solvent. They can denature your protein. Or sometimes you can use this thing called urea, okay? Urea is a chaotropic agent which disturbs or disrupts hydrogen bonding between water, okay, and it's going to lower the hydrophobic effect. Okay, so a big part of why your protein can fold, right? We say the core of the protein is mainly hydrophobic. Okay, a large reason for that protein folding is because of hydrophobic effect. Okay, and remember, hydrophobic effect is due to water-water interaction, right? Okay, now if you have a thing like urea, urea is going to make hydrogen bonding with water, so you have less water-water interaction. Then as a result, the hydrophobic effect will go down. Okay, so you can actually have more hydrophobic residues exposed to the solvent. Okay, so that's one thing that you can use to denature a protein. Okay? And in fact, urea, if you guys have done biochemistry experiments, urea is used a lot of times to solubilize proteins. Okay, so a lot of times when you are doing recombinant protein production, your protein is insoluble. Okay, you make an insoluble inclusion body, we call that usually. But inclusion body is not good for whatever that you want to do with your chemistry. So you can add large amount of urea and then you can solubilize it. Okay? Now, some of the things that you see when proteins are denatured, give you a picture or two pictures of egg, right? This is egg in its natural state. Okay, so it's slightly transparent, it's very liquidy. Now you add heat to it, okay, obviously it becomes solid. Okay, so you know this, you've been doing this for forever. Why does it do that? Okay, because the 3D structure of your soluble protein is destroyed, okay, and then your proteins start to form aggregates, and then it becomes essentially solid, okay? So this is a process called denaturation, right? So you can actually take egg white, okay, this guy, you know, the egg white here, and then you take it out, right? And then you dissolve it using urea, and then gradually take urea out, and then you can fold the egg back, okay? So protein folding, right? So then a real question that we should always ask ourselves is that how do proteins know what kind of secondary or tertiary structures to form, okay? We saw in our previous lecture that they form alpha helix, they form beta sheets, whatever, right? Turns out, okay, it really turns out that most of the information for determining the three-dimensional structure of the protein is carried on the amino acid sequence, okay? So what that means is given a particular amino acid sequence, there's usually just one way that it's going to form into a major structure, okay? So it will not like fold into different structures, okay? Given in the same physiological condition, it will generally turn you this particular structure, okay? So three-dimensional structure is sort of determined by the primary sequence, okay? Now, why is that? Okay, because the primary sequence tells you what kind of amino acid residue is going to be at what position, right? And then these R groups can form hydrogen bonds, can form hydrophobic interactions, right? So then, of course, it's going to form a most likely structure, okay? So here's a very famous experiment done a long time ago by Chris Engenson. This is the RNase, okay? RNase. Right, so RNase is an enzyme that degrades RNA, and for your information, RNase is like almost everywhere. Okay, so if later on when you guys go and do your undergraduate research or research in general, if you work with RNA, you have to be careful because RNA it degrades very easily, especially because RNase, okay, the enzyme that degrades RNA is very stable. And you got RNase on your fingers, you got RNase like everywhere, okay? So if you are not working in a very clean environment, your RNA degrades like crazy, okay? So RNase is quite stable. Because it's quite stable, they decided to use this enzyme and to look at its process of denaturation and refolding, okay? Now, interestingly, what they can show is that denaturation and refolding can be reversible. Let's take a look. You start with the native molecule. This is the RNase. And in this particular RNase, what you notice is that it has a structure, and obviously it's shown in squiggly lines, so whatever. What you also see is you see these disulfide bonds. These are disulfide bonds, okay? And we'll talk more about disulfide bonds later. So what they can do, okay, to denature this protein, the first thing is I'm going to reduce away the disulfide bonds using this thing is called the beta mercaptal ethanol. Essentially, it's just this. And then I'm going to use urea, like we just talked about. So beta mercaptal ethanol can reduce the disulfide bonds. So we're going to essentially turn them into SH. So separate disulfide into SH, right? That's what we're going to do. And then urea disrupts the hydrogen bonding between water, so then your protein can be exposed, and then it becomes soluble, even if it's denatured. Okay, so this is the denatured molecule. We're gonna gradually remove the urea. Okay, so as you're gradually removing urea, then the protein is going to start feel the hydrophobic effect again, right? And then the hydrophobic effect is going to cause the molecule to have the hydrophobic cores in the middle, right, in the core, the hydrophobic residues going into the core, and then the hydrophilic residues be on the outside. So it's going to be folding back to its native state, okay? And then once it's sort of folded back into its native state, we're going to add oxidizer to regenerate your disulfide bond. And then they were able to show that this is a completely reversible process. You can reduce, denature, and then you can wash away the urea, oxidize it, and then you can regenerate the original structure. That's pretty cool, okay? What they noticed was that what if today, instead of removing urea first, we do the oxidation first. So here, obviously, step 1, remove urea. Step 2, oxidize. What if we reverse that? We do oxidation first and then remove the urea, right? If you do the oxidation first, you're going to cause the disulfide bond formation between random cysteines, okay? Why is that? Let's say, originally you're supposed to have cysteine 40 with cysteine 95. Supposed to be cysteine 40 and cysteine 95 form a disulfide bond. But after you denatured the protein, okay, and it's floating around, and then you immediately add the oxidizer, okay, the cysteine is just going to find whichever other cysteine, and then it's going to form a disulfide bond. For example, cysteine 40 can interact with cysteine 26. They form a disulfide bond. Or 40 can interact with 58. They form a disulfide bond. Can interact with cysteine 84, form a disulfide bond. Okay? So then, what they showed was that incorrectly folded proteins due to random disulfide bond formation. Okay? Only one of these molecules are active because given 8 cysteines, you can have about 105 different combinations. Okay? So if they do oxidation first and then removing urea second, then their protein is likely to become a garbage protein, okay, a misfolded protein, because your cysteine can easily oxidize and form the disulfide bond with another cysteine. Okay, but in the first, in the case of the left side here, it's harder because you remove the urea first, okay, and then the protein has already become a structure that looks like the native state, and then you oxidize, then it will find whichever cysteine is closest to each other. Okay. Regardless, the conclusion is that protein folding is reversible. That's pretty cool. Didn't think you can refold an egg. Maybe you guys should try that at home. Okay, take an egg, fry it, and then put like urea on it, dissolve it, see if it can refold it back. Okay, denaturation of proteins can be actually monitored. They had to know, right? How do you know if it's folded? How do you know it's unfolded? So there are some things that we can do. This is the thermal denaturation of RNA. Okay, so you don't use urea or anything like that. You just use temperature to denature your protein. Okay, so we know that proteins are sensitive to temperature change. Okay, so the difference between the native and the denatured conformations can be detected by several spectroscopic methods. Okay, you can use spectrophotometric measurements, for example, at specific UV absorbance. Okay, or you can see how you have a larger change in optical rotation, or you can see how the solution's viscosity can increase due to the denaturation of your protein. Okay, why do you think the solution's viscosity change? 你觉得为什么 viscosity 会变？如果你觉得 viscosity for soluble protein is higher 还是denatured protein is higher? Probably denatured protein is higher, right? Because as your protein is denatured, it becomes more of a polymer-like structure. When it's native state, it's globular, it's very soluble. but then when it's becoming denatured, usually it's longer random coil, right? You have a coil-like structure, so then as a result, the viscosity of your solution increases. Makes sense, okay? All right, so using all three of these techniques, you can observe the fraction of the protein's denaturation. Okay, so in this case, they took RNA, and then you look at as a function of temperature. This is the fraction unfolded. Okay, and you can see that you have a huge change between about 25 to maybe 35. Around this range, you have a large amount of protein denatured. Okay, so just to show you that proteins are sensitive to heat and you can measure it using many different methods. Now we know that proteins can be denatured in response to heat, in response to addition of solvents or chaotropic agents like urea, in response to adding, changing the pH. Okay, so let's take a look at how is it that protein folding is a natural process. So we're going to come out and say that folding of globular protein is thermodynamically favorable. And you're just like, what? Okay? You just love to hear the word thermodynamics. Okay? The folding of globular protein is clearly a thermodynamically favorable process under physiological conditions. Why? Because otherwise, your proteins will all be denatured, right? Because like you're living at this moment with soluble proteins, meaning that your proteins, between the protein in a denatured state to a natured state, okay, to a folded state, properly folded state, is a thermodynamically favorable process, okay? Because if it's not, you will not be standing here, okay? You'll be a big blob of proteins. OK， 这边说 thermodynamically favorable 是什么意思？调查机是负的嘛 ？OK， 就是说在热力学的角度上来说，这件事情是会发生的。OK， 那为什么会发生呢？为什么是一个非常 obvious？OK， 就是说就是一个你连想都不用想，这句话一定是对的。原因是因为你看哦，你现在人坐在这边，对不对？啊，你身体里面有很多 soluble 蛋白。OK，soluble 的这种 globular protein， 例如像什么 myoglobin 啊、hemoglobin 这种东西 ，OK， 那它们都是这些 globular folded 的 protein， 对不对 ？The fact that 你会身体里面会有这些 protein， 就代表说在 physiological condition 下面呢，这些 protein 的 folding must be 一个 thermodynamically favorable， 因为它如果不是的话，你的 protein 就会是乱七八糟的东西。那你就不可能坐在这边，你就会是一坨蛋白 ，OK， right? But you're not, right? So folding is thermodynamically favorable. We have to accept that, okay? It may not be the most thermodynamically favorable state, okay? But it is thermodynamically favorable for the protein to fold, okay? Now, what that means is that the overall free energy change for folding must be negative. Okay, this is saying that the delta G of this process is less than 0. Okay, basically that's what it means, right? The energy for folding must be negative in order for this process to occur. Okay, and this negative free energy change is achieved by a balance of several thermodynamic factors. Okay, you need to know each of the individual factors. Right off the bat, I'm going to tell you protein folding. Okay, folding of proteins is against entropy of the protein molecule. Does that make sense? Okay, why? Because if you think about it, a protein, okay, let's say you have a protein that's 300 amino acid residues, right? If you were the molecule, you are the 300 amino acid long protein. You want to be able to rotate like crazy, if in terms of entropy, right? Entropy always wants to move like crazy, okay? However, when that 300 residue protein becomes a folded protein, you restrict rotation of many different bonds, okay? Now, you're a protein and you're just like, what? I don't like that, okay? So entropy of the protein goes down. If entropy goes down, delta G goes up or down? Delta G goes up, right? Delta G becomes more positive, right? So in terms of entropy, entropy does not favor protein folding per se, okay? Now, we're going to see some other stuff. You are going to gain energy from where? From charge-charge interactions. We know at physiological pH, you have acidic groups like glutamate, aspartate. You have basic groups like lysine and arginine, right? That one is going to have a negative charge, the other one is going to have a positive charge. So if they're close together, they form a very good thing called salt bridge. So when you have COO minus and NH3 plus, this guy is a salt bridge, okay? It's a salt bridge. One is minus, one is positive. Together, they're very happy, okay? So having this electronic interaction makes it very happy, okay? So this will provide delta G go down. This will provide delta G go up, okay? You can also have internal hydrogen bond formation. You can have stabilizing hydrogen bonds between polar R groups like serine and another serine, okay? So you can have, for example, like that, okay? So you can have something that works like that, all right? Hydrogen bond formation also cause your delta G to go down, okay? Because formation of hydrogen bond is favorable. You're going to have van der Waals, okay? Van der Waals, sure, you can have stabilizing attractions as globular proteins are well packed. So what that means is you can have phenyl rings next to phenyl rings, okay? So they have attraction for, all of them are very tightly packed van der Waals. Having van der Waals attraction also lowers your delta G, formation of van der Waals. Pretty cool. Hydrophobic effect. Hydrophobic effect is when you have all these hydrophobic things cluster together and the water is sort of just outside. You minimize the surface area between hydrophobic and hydrophilic. Okay, so remember that this is minimizing hydrophobic and hydrophilic surfaces. Okay, having hydrophobic effect, you always lower the delta G. Okay, so you have 4 characteristics. That's going to lower your delta G. You only have one that increases your delta G. So overall, delta G lowered. Okay, so these characteristics sort of governs your protein folding. 当你今天如果这一坨啊是 hydrophobic，OK， 我们说这个是 hydrophobic，OK， 当你今天有 hydrophobic 跟水 ，OK， 跟水有一个界面的时候 ，OK， 那这个界面呢，你的水呢会呈现一个非常 regular 的一个像 crystal lattice 的一个 structure，OK， 那这样子的话，你水的 entropy 会往下掉 ，OK， 因为水它也想要流动嘛，对不对？那问题是说，如果你有 hydrophobic 东西在那边的话，水的 entropy 会 drop， 那不 OK， 对不对？所以 hydrophobic effect 就是说，自然会 process， 就是说自然而然就会把 hydrophobic 全部集中变成一颗圆的，降低 surface area，OK， 那这样子的话，你的 hydrophobic 跟 hydrophilic 的 interface 就会降低，然后你的 hydrophobic effect 就会比较明显，这样。对，主要是为了要提高水的 entropy， 这个跟那个 protein 的 entropy 不一样哦，不要搞混哦。对，就是当我们在讲 hydrophobic effect 的时候，我们在讲的是水的 entropy，OK， 但是跟 protein 的 entropy 不一样。In this case, in protein folding, protein's entropy drops, but water's entropy increase. Okay, that you have to make clear. Okay, cool. All right, so this is like very important. All right, so if we summarize it into an energy or a little schematics, so here we're only looking at minus T delta S, meaning we're only looking at entropy. Okay, so here we only look at entropy. This is minus T delta S from the decrease in conformational entropy. So this is of the protein, okay? and you see a large increase in delta G for protein folding. Okay? Why is that? Because protein is now restricted in a specific shape. It doesn't like that. Okay? So, delta G increases. Why? Because delta S decreases. Now, a couple of things. In this folding process, you also get what? You also get hydrophobic effect. So this hydrophobic effect is essentially from water. So having a folded protein minimizes the hydrophobic and hydrophilic contact surface so that you have a decrease in energy, in free energy, due to hydrophobic effect. However, overall, you still have a net positive. Okay, so this is a net positive for folding. Okay, so based on entropy alone, protein folding is not good until you have the enthalpy term. Okay, enthalpy term for folding is favorable. Why is that? Here we have salt bridge, we have hydrogen bonding, we have van der Waals, okay, etc. We have good attraction between the protein intracellular. Okay, so this is all talking about intramolecular interaction. So we have all those good interactions between the protein itself, and then it's going to give you a large negative delta H. Okay, so you add this guy and you add this guy together. What do you have? You have a negative delta G for folding. Essentially, the stability of the folded structure of a globular proteins depends on the interplay of three factors. The unfavorable conformational entropy change. So this is talking about proteins entropy will decrease. However, you do have favorable enthalpy contribution from intramolecular interactions. Okay, now this is important. Intramolecular is within protein itself. Okay, so within the protein itself, that's what intra means. Intra is within itself. And then you also have a favorable entropy change arising from the burying of hydrophobic groups within the molecule. This is the hydrophobic effect. So let's get this straight. Okay, so unfavorable due to a drop in entropy of the protein, but favorable due to intramolecular interactions, salt bridges, hydrogen bond, van der Waals, and then a favorable hydrophobic effect. And then you have a folded protein. Now, disulfide bonds. Okay, don't forget disulfide bonds. Disulfide bonds are interesting in a way that they are not essential for refolding, as we demonstrated in the RNase experiment. So disulfide bond formation is not super important for getting the three-dimensional structure of the protein. Okay, that is determined by what? By the amino acid sequence. Okay, so it folds to its preferred state. Now, but what the disulfide bonds can do is that they provide significant stability to the structure once it's folded. Now, in some cases, like insulin, for example, insulin has a couple different chains. Okay, you have the alpha chain and the beta chain. In those cases, disulfide bonds are used to keep them together. Like, for example, in your antibody, okay, antibody has that small, V part and then the long part, right? The light chain are held together by disulfide bonds. Okay, so disulfide bonds are also used in that sense. You can have two separate molecules physically connected using disulfide bonds. Okay, it can do that as well. But for the majority, what disulfide bonds can do for you is it offers stability for your structure. Some proteins are stabilized by internal disulfide bonds in addition to non-covalent forces. So what I want you to look at is this particular graph right here. So this is a protein called BPTI or BPT1. And essentially I want to show you this is native protein and this is after removing one disulfide bond. As you can see, the thermal stability of this protein is a lot better in its native state. How do I know that? Because after increasing the temperature to 60, I have only maybe about 10% denatured. for the native protein. But if I have one disulfide bond missing, then at 60 degrees, I'm looking at about 60% denatured. So what that tells you is that if you don't have disulfide bond for this particular protein, that its thermal stability decreases. Okay, so with a little bit of heat, and then your proteins like vibrate, and then it breaks. Okay, having the disulfide bond, you can think of two proteins, right? Well, one protein, and it's moving, but you have a disulfide bond in the middle, and then you stick it together, and then it's happy. Now, couple things about disulfide bonds, okay? Couple things. Turns out disulfide bonds are relatively rare and are found primarily in the proteins that are exported from the cells, such as your RNase, insulin, and other proteins that are meant to do cell-cell talk, okay? They're exported. One potential explanation is that the environment inside of the cells is reducing. By reducing, it means that it favors reduction. Okay, so it favors having electrons on them. Okay, so this is inside the cell. Okay, most cells inside the cell is more reducing than oxidizing. Okay, however, outside of the cell can be more oxidizing. Okay, how do we know that? Especially in animal body. You know, we breathe a lot of oxygen, okay, and then, you know, our body is pretty oxygenated, right? So outside the cell, you don't have enzymes or proteins that help to maintain the reduction state. So then outside the cell is usually more reducing. Okay, so for example, if you guys are interested in doing protein expression of an enzyme or a protein that have disulfide bonds, okay, and you want to do that in E. coli, use E. coli to produce that, usually we will have the protein folded in the periplasm. Okay, so not inside the cell, actually outside the cell. Okay, you'll have the protein folded outside the cell. Why is that? Because outside the cell is more oxidizing. Okay, so if you want to have a disulfide bond protein folded, okay, you usually do that instead of doing it inside. Okay, make sense? So internal environments, in the most part, are oxidizing and stabilizing the bridges. Okay, so this is disulfide bond. So now we're going to talk a little bit about the kinetics of protein folding. You would think that in order for a protein to fold into a correct secondary and tertiary structure, it would take a long time. However, it turns out it's not a long process. Why? Because all these are chemistry things, you know, it obeys the laws of physics. So it's actually relatively remarkably rapid. Okay, so the folding of globular protein from their denatured conformation is a remarkably rapid process, often complete in less than a second. Okay, so for example, E. coli can make about 100 amino residue polypeptide in about 5 seconds at 37 degrees. So you actually produce proteins pretty fast. Now, this is a key concept. Protein folding is not a completely random search through a vast conformational space. And by conformational space, essentially it means that you have a protein, okay? Like initially you have a protein, okay? And then you want to fold. And there are many different ways you can fold a protein, okay? So if you take all the possible folding, okay, you can have like billions and trillions and 10 to the, I don't even know how to count, okay, number of different conformations. Now, protein folding is not like that, okay? It's not a completely random process, okay? After you fold a little bit, immediately you don't go to the other random states, right? So you'll see in just a minute. This is called the funnel theory. A rapid kinetic study using a variety of physical techniques to monitor the different aspects of protein structures, we can show that folding takes place through a series of intermediate states. Okay, so essentially what this is saying is after the protein goes from unfolded state to an intermediate state, then let's say you have maybe a lot of different states on top. After you chose this particular route, you don't go back up, okay? You don't just go back to the random states. You don't, you can't. Okay, so as a result, the more folding you do to the protein, you actually restrict the number of conformation it can have, which makes sense. After you folded an alpha helix, it's very difficult for the alpha helix to all of a sudden become a random coil or all of a sudden become a beta sheet. Why is that? Because the formation of alpha helix is very favorable, right? You have hydrogen bond, so it's stabilized. Unless you have energy input to kick it back up, it won't. A simplified representation of folding pathway of a protein is like this, okay? You have U unfolded, I intermediate, and then you have a final step of folding, a completed folded state, okay? Folded or native. So you go from unfolded to partially folded to more folded, blah, 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 and then eventually to completely folded. During the folding, sometimes you can go into the off pathway states, okay? So these are essentially the unfolded, okay? Unfolded, misfolded states. Now, a lot of these unfolded or misfolded states, some of them are reversible. A lot of times, they're not reversible, okay? So if they're not reversible, then they form aggregates, which then essentially your protein folding process reach a dead end. Okay, so this happens sometimes if you're expressing proteins maybe too fast, okay, or not in an environment that is favorable, then it's easy to get from the intermediate state into a messed up state. Okay, so we usually just call that off pathway states. Right, so that happens. And we'll talk about some of the things that we can use to go back, but just bear with me. There are several different off pathway states. One very good example is prolines, cis, and trans conformation. Okay? Folding can be delayed by trapping of molecules in the off pathway state. Okay? In this particular case, the isomerization between trans and cis can happen given that you have an enzyme. Okay? So, one of the most common folding error results from the incorrect cis-trans isomerization of the amide bond adjacent to a proline residue. So that means that during translation and during folding, sometimes you can actually have a trans to cis flip. And we know that most proteins, okay, most peptide bonds are in the trans state, not the cis state, because trans state is more favorable. However, in this case, okay, you do have a 4-to-1. So you do have a little bit of misfolded probability. Now, if your protein becomes misfolded in this particular case, you have to use an enzyme called the proteoisomerase that will speed up the conversion. So you go from cis back to trans, and then you can continue to fold. In this case, this would be a reversible off state, okay, off pathway state, because you can actually use an enzyme to pop it back in, and then you can continue the folding. Folding funnel model. So this is what I was talking about. As protein is folding, it follows a downhill trajectory on the energy landscape. So you can think of this as energy landscape. So increasing in energy, okay? So when the protein has a lot of energy on top, when the protein just after translation and it's unfolded, it has many different states that it can adopt, many different conformation. It can be beta sheet, it can be alpha helix, it can be whatever, random coil, whatever, okay? And as it's folding, gradually you're folding and folding and folding, and then you're folding into its closer and closer to its native state, to its true and most stable state, when it's becoming folded. So as you fold a little bit, you see how you no longer have the space to go and enter a different random coil structure. So the more you fold, the fewer conformational state that your protein can have, which makes sense. Once you form a stable secondary structure, for example, you form an alpha helix. Alpha helix don't just like randomly pop out. So gradually you converge down to your completely folded state. Now, in the middle, in this globular state, we usually call that a molten globule state, and it refers to partially folded states that have some native-like secondary structures but lacking defined tertiary, meaning that in here, before it reaches the final state, it has formation of alpha helix, beta sheet, but then you still have movement. Okay, you still have different conformation and then different topology. Okay, remember we talked about tertiary structures. We have topology, right? That CATH category thingy, right? The cath, right? You have different topology, even though you have already the alpha helix and the beta sheets. Okay, so you can still have different topology moving in here until it eventually becomes the final state. Now, obviously, you should recognize that protein folding is not going to be an idealized funnel, okay, because you will have states, localized states that have higher energy. So in reality, what this does is it does not give you a random folding process. Let's say you start from here, you're folding and then you hit here, and then gradually you find whichever is stable, and then you follow the stable route until the end. So you don't have the large conformational possibilities as a completely random process. So this is not a completely random process. So after you fold a little bit, and then you sort of follow that direction. Now, sometimes you get to these meta-stable intermediate states. Okay, these are intermediates in protein folding. Okay, so remember we went through the exercise of what is stable, metastable, and unstable, right? So anything unstable is something that you cannot trap. It just spontaneously becomes something else. Metastable is like an intermediate that you can trap, but it's not the final product. Okay, so here you do have chances of forming these intermediates. Okay, they are not at the lowest in energy, but they sort of become locally stable. So you can actually get these local structures. Of course, they are metastable because given a little bit more energy, they can come out of that well and then become the final stabilized structure. And then show you a little video about that. All right, so this is initially a random coil right here. You have a couple amino acids, right? And then this is the energy sort of contour map. Right. And then it's folding, so you're initially surveying on top, and then you see how it randomly rotates. And all of a sudden, you start to see formation of your alpha helix, and then gradually it enters the lowest in energy, which then you see beta sheets, right? So this is beta sheet, 1 beta sheet, 2 beta sheets, 3 beta sheet, beta strand, okay? 1, 2, 3, 4, and then 1 alpha helix. Okay, so as you can see, initially it's like rotating like crazy because it has a lot of energy. And right after it starts to fold, that folded structure no longer disappears. Okay, and then that's it. Okay, so then you fold and then you end up with a complete folded structure. Okay, because the formation of a secondary structure is rarely disrupted. 懂我意思吗？哎，你 form a secondary structure 之后，它就 secondary structure， 它就很稳定，然后很开心。你不会没事把 secondary structure 又变成一个 random coil。OK， 所以当它形成这些不同的 secondary structure 之后，那就只是这些 secondary structure 在 rotate， 直到它达到一个最低的能量阶的一个 state。So hopefully now you know how protein folding, like what are the sort of the principle that drives protein folding, right? So that you should know, right? It's the interactions and stuff like that, right? And you saw that protein folding is not completely random. That's fine, okay? So the next thing I want to show you guys is protein topology and folding rate. Okay, so topology, again, goes back to our Friday's discussion about how you can have a lot of different alpha helix and beta sheet, and they can assemble into different topology. So topology is kind of like the the tertiary structure, the structure of this particular domain. Okay? First thing I want to show you guys is if you look at the figure on the right and you look at A and B, they are two different proteins, right? But they have the same fold, okay, with the same domain. Okay, so essentially, it shows you that topology, okay, with this kind of folding domain thing, this structure, this tertiary structure, may not be always related to the exact function of this protein. Okay. You can have different enzymes that do different things, and they have similar folds, okay, the similar tertiary structure. You can actually have that, no problem. And here, what I want to introduce to you guys is that topology is an important factor in determining the rate of protein folding. So the formation of alpha helix and beta sheet, that's pretty fast, no problem. Okay, now, the difficulty comes in is when you assemble the different secondary structures into their tertiary structures. Now, for example, I'm going to give you this idea of contact order. Okay? Contact order is like sequence separation between residues that physically contact in the native state. Okay, so essentially you can think of it as this. A protein that has low contact order is when this little ball right here and another ball that has physical contact with each other, they are close to each other. Something that has high contact order, then is opposite. Okay, so this protein goes like this, okay? Like that, comes out, and then like that. So between this little circle and this little circle, essentially it went through like that and then comes back. So if this guy is 1, 2, 3, 4, 5, if this was number 5, then 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. Whereas, if we're talking about, for example, this one and this one, or we take similar position. Let's take those two. You have 1, 2, 3, 4, 5, 6, 7, this is 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19. You see how they can be further apart, okay, in terms of their primary sequence. But when they become folded, they are next to each other. Okay, this is called contact order. Okay, so turns out proteins or polypeptides with higher contact order will fold slower. And that makes sense. Why? Because if you think about it, after the formation of your alpha helix and beta sheet, if the two helices are next to each other, then they immediately touch each other very fast. But if the two helices are really far apart, then for them to come together and touch each other and become the folded state, it takes more time because they have to go rotate, 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 and then rotate, eventually contact each other. Make sense? Okay, so if you want to have a protein that folds really fast, then you generally want to have a lower contact order. Okay, so because immediately after they fold, they rotate a little bit and then immediately they contact and they form the topology that you want. If they're further away physically, then the whole molecule has to rotate and spin until they touch each other and then form whatever structure they form. It makes sense, right? It really actually makes sense. Because if they're further apart physically, then it takes a longer time for them to meet each other. Right? So protein topology decides the rate of the overall folding. Because some topology may be difficult to get to. Because it requires this alpha helix and this beta sheet to come together. It takes longer time. So sometimes, we're going to get back to the idea of protein misfolds. So while most or many, if not most, proteins can fold themselves, some proteins actually require the actions of specialized proteins called molecular chaperones. You guys know what chaperones are? 那种古时候有没有女生要去舞会的时候，你会有个 chaperone， 一个男的陪你去参加舞会，那个东西叫 chaperone， 就是说 chaperone 就是那种陪伴的一个陪伴的人或者是一个东西这样。Chaperone protein, same thing. Okay, you have a chaperone meaning that this protein, you have a chaperone person that protects you in a way, or that guides you to the place that you're supposed to be. Okay, 那种东西叫 chaperone. Okay, so molecular chaperone is a protein that guides the protein that's misfolded to become folded. So molecular chaperones, they function to keep the newly formed protein out of trouble. Trouble meaning improper folding or aggregation. Okay, so sometimes if your proteins are folded too fast, it gets trapped into the off-pathway state, misfolded. Then you got this molecular chaperone that's going to come in, take you out, and then give you some energy, make you refold. Okay? Now, some improper folding results from intermolecular contacts. Okay, so what that means is a lot of times, let's say, you have a peptide. Here it's hydrophobic. And supposedly it's going to fold like this. Okay? And then it's going to be on the inside, hydrophobic. But sometimes, if it folds too fast and too excited, it becomes two hydrophobics come together, and three hydrophobics come together, and then they become a clump. They aggregate, okay, if it's made too fast. So before your protein can fold, they aggregate, precipitate, and then the protein becomes non-functional. So for those of you that work in the molecular biolab, we see this a lot. So through evolution, this actually happens in nature too. Okay, so you don't always have to see this in engineering, but you also see this in nature. For example, a lot of times when your cells are heat shocked. Okay, so like we make our proteins very comfortably at 37 degrees, but what if all of a sudden your temperature goes up to 42 degrees? Okay, so cells will have to develop some kind of a mechanism that gets the unfolded proteins out of their miserable shape. Okay, these things are then called molecular chaperones. Okay, so let me first go over this little picture. So after the protein synthesis, this is the U state, unfolded. You want to have a folded state. There are some things that could happen. For example, you can have disordered aggregates, so the unfolded proteins just precipitate together, like that. Or you can start folding into intermediate states, start to have some kind of domain, and then the domains aggregate, and then it's still aggregate, same thing. Or they can be degraded into fragments, and then the fragments could aggregate. Or you can keep on folding and folding, and then you fold domains. Okay, domains, they become folded and they form oligomers, etc. Okay, now if you look at the energy contour on figure B right here, what we saw on the previous slide was essentially this. That's what we saw. But if you give it more energy and you enlarge the overall landscape, you can have intermolecular contacts. So what we saw was intramolecular. Intramolecular is talking about the foldings, the molecular foldings by itself. Makes sense, right? Now, when you have multiple of this particular protein, they can have intermolecular interactions. We're not even talking about quaternary structures. This is not quaternary structure. This is just crazy intermolecular interactions that precipitate them into something that you don't want. Things like amorphous aggregate. Okay, just a bunch of random crap that aggregates, no function. You can have formation of oligomers, no function. Or you can have amyloid fibers. We'll see amyloid fibers when we talk about the mad cow disease. Yeah, that's not good either, okay? So those things a lot of times come from intermolecular interactions, and too soon. Okay? So we will have intermolecular interactions between proteins, but not before they're folded. So this is the structure of a molecular chaperone GRO-EL and GRO-ES. It's called GRO-E. You have S and L. S stands for small, L for large. Okay, so GRO-EL is the large component. ES is the small component. So I'm not going to go into the detailed mechanism of how this thing works, but I'll give you a pretty good picture of how it works, okay? So if you look at this protein, it's a huge protein that looks like a little trash bin, okay? It's like a can, okay? So what do? You have a misfolded or unfolded protein like this, and it's unfolded. And what your GRO-EL and GRO-ES can do is it's going to grab the unfolded protein into your GRO-EL, and then you can line up the hydrophobic residues. You can line them up, okay, so they interact. They have very good interactions between the chaperone and your protein. And then what do? You load ATP, the magical power of ATP. Remember, ATP has a lot of what? Energy, right? Has a lot of energy stored inside. So ATP binds to your molecular chaperone, and when ATP is hydrolyzed, your molecular chaperone changes shape. You have a conformational change. GRO-EL changes shape. Initially it's a bracket, now it's like an ugly bracket. And then this shape change will cause the hydrophilic residues to be exposed. So then that's going to change the interaction between your protein and the molecular chaperone. After it's folded, then your folded protein can then leave the molecular chaperone. Okay, so actually this is overly simplified, okay, by the way. But the basic idea is this, okay? You have a molecular chaperone, and then you take in the protein. Use ATP to change the conformation. Your protein becomes folded, and then the protein comes out. Okay? So you use ATP to assist your folding. How does it do that? Okay? So how does it do that? If you go back to your energy diagram here, in a way, you can sort of think of it as like, okay, so your misfolded protein is like here. It's misfolded. And you cannot just unfold a protein because it's at an energy minimum. So how do you get it out of the miserable hole? You give it some energy. Okay? So you throw ATP inside, essentially. Okay? So you elevate the energy up, and then it can fold. So in a way, that's sort of the logic behind it. So turns out many of the chaperone proteins are also heat shock protein, which help to refold denatured enzyme upon increased temperature. Because we saw that increasing temperature will denature some of your proteins, right? So a lot of times, like E. coli has heat shock proteins. Okay, so like when you heat shock E. coli, meaning you throw E. coli in elevated temperature, some of the proteins will misfold. And then what E. coli will do is E. coli will express heat shock proteins, and then these heat shock proteins will help to refold your proteins. So it's kind of like that. Protein misfolding. Protein misfolding is a huge problem, especially in humans. Okay, so if you have protein misfolding, it's going to cause diseases. Okay, it's going to cause you to have problems. Okay, and protein misfolding is the basis of several diseases, including Alzheimer, including Parkinson, and these things are what? Neurodegenerative diseases. So they're the diseases or problems that causes memory loss, that cause you paralysis, that cause you all sorts of different problems, okay, in human physiology. So in humans, most of these diseases are associated with the formation of highly ordered protein aggregates called amyloid fibers or plaques. Okay, so usually we call them beta amyloids because they're rich in beta protein or beta sheets. Okay, so on the left here, you can find different diseases. These are all the different diseases that humans can have and what are the proteins associated with them. Okay, in particular, you can have Alzheimer. Alzheimer associates with amyloid beta, beta amyloid, essentially this. And we'll see in just a minute the formation of beta amyloids. Okay, these are highly ordered protein aggregates. So essentially, they insolubilizes your brain almost. So this is amyloid fiber. Amyloid fiber, you can take EM and then you can actually see it. Okay, you can actually see this fiber. And this amyloid fibers are made out of repeating beta sheets. So if you take a top-down view on top, this is what it looks like. So it's very repetitive beta sheets, and beta sheets are very, very stable. It's a very stable secondary structure. And when you generate large structures out of this, you can no longer use chaperone proteins or anything like that because this is a macro structure. Okay? It's not a small enzyme or a small protein. This is a macro structure. And the formation of this will cause you, essentially, you can think of your head, okay? Your brain initially is filled with different soluble proteins and stuff like that, okay? But the formation of these proteins will cause your head's protein to come together and essentially precipitate, okay? So make aggregates of proteins inside of your head. So your head becomes sponge. Have you heard of that? Your head becomes sponge form. 就像会变像海绵一样。为什么叫海绵？因为你的头里面就会有洞，你的大脑里面会有一堆洞。因为你的那个大脑原本是很多 enzyme 嘛 ，okay? It's filled with stuff, okay? And then when the proteins start to aggregate, your brain starts to form like a sponge, okay? Because things start to aggregate, and then you have holes inside your head, okay? And then obviously that's gonna have problems, right? It's gonna cause you to, In particular, I want to talk a little bit about prions, or some people call it prions. Prions are proteinaceous infectious particles. Prions. Have you guys heard of this? Yeah, it's quite scary because there's nothing we can do about it. Absolutely nothing we can do about it. Prion diseases, in general, they cause neurodegenerative diseases, just like Alzheimer, Parkinson's. All of these problems are all associated with the fact that you form crap in your brain, okay? You form aggregates, insoluble aggregates inside of your head, and then they interfere with your neurons talking to each other. Prions, okay, they are essentially proteins. This was actually a very difficult thing for science because we know that DNA is infectious, okay, like bacterial infection, viral infection. So we know that DNA, RNA could be infectious. We didn't know that proteins could be infectious as well, but until we saw that we have these prion diseases, okay? Essentially, what it is you have prion-related proteins. We're going to call them PRPC. These are normal proteins found in cell. You have them, I have them, we have these proteins, okay? We have them. They're natural proteins inside of you. Now, PRPC has a small chance, nonetheless, a chance to spontaneously form PRPSC. Essentially, we call them the infectious prion. Okay, so they are a specific structure of this peptide that is filled with beta sheets. Okay, so if you look at this is PRPC, you see mostly alpha helix. PRPSC, mostly beta sheet. Okay, and beta sheets, from our discussion a couple weeks ago, we know that beta sheet can pack very well. Okay, they have very good packing, stacking. And because they have very good stacking, they form these beta amyloids. Okay, they form insoluble stuff. Okay, now what's interesting about prions is that once you have PRPSC, okay, these prion proteins, they can go and interact with the prion, the normal prion proteins, the prion-related proteins, the normal proteins, and then you're going to turn them into prion proteins. Okay, so you can think of, you have primarily this, and you have a little bit of this guy is going to come touch the PRP-C and then turn them into PRP-SC. That's scary. Now, what's even more scary is that your PRP-SC, your prion proteins, are super stable. They can tolerate very high temperature. You can like throw them into autoclave and then they'll still be there. Maybe I'm exaggerating a little bit, but they're very stable. Okay, so you cannot kill them with heat. Prions, they cause mad cow disease, scrapies in sheep, and then cruise in human. Do you guys know what cruise are? Cruise happens to people that eat people, cannibals. Okay, so like in the really, really poor countries in Africa, they eat people, yeah, and then they turn kind of psychotic. That's what cruise are, okay? Yeah, it's a neurodegenerative disease, okay? And that happens because they eat like brain tissues of other stuff, okay? People, for example, and then they make them crazy, okay? Like rabies, kind of related to these things, okay? They have that kind of behavior, all right? Yeah, so mad cow's disease, okay? So why are we so worried about mad cow disease, okay? It's because, you know, this is different from virus or bacteria. You know, like bacteria and virus, you're like, yeah, just cook your meat like completely cooked and then you're fine, right? That's how you kill bacteria or virus particles, like water, you boil it. But the problem with prion is that like you can cook your meat as much as you want, but then you can still have populations of your PRP-SC because they're not degraded by heat. And then you're eating and you're eating and eating. You eat small amounts of these proteins, and then you're fine now. But 20 years later, you're going to turn, you know, mad cow. Okay? So that's why people are scared. Okay, so I'm going to show you some videos related to prion diseases. So this is the aggregate. So then your brain will become sponge form. So then you literally have holes in your brain. Virtually indestructible. That means that you can't kill it. Okay? So that's that. And then I'm going to show you more scientific one. So prion disease. This one doesn't have talk, so I'll just have to like narrate. But you can follow the video a little bit. Okay? So a lot of these, not necessarily related to prion diseases, but like all of these neurodegenerative diseases, these things are really difficult to deal with. Okay? And hold on, I'm just going to backtrack a little bit. These are the main symptoms. You have social withdrawal. Insomnia. Insomnia is you can't sleep. Dementia. Dementia is like you go crazy, okay? Muscle paralysis. Muscle paralysis. Paralysis, you know, 麻痹, right? Paralysis. Coma. That's the worst thing that can happen. You're just like, you know, you're just there. You're alive, but you're not awake, okay? So all of these are related to neural systems, obviously, okay? These are the main symptoms related to prions, okay? And all of these are caused by tiny protein particles called prions, okay? And you can contract this disease by consuming material from animals infected with scrapies prion forms, like mad cow, okay? Now, a lot of people are going to tell you that, which is why you should not eat like cow bone marrow, like bone material, because obviously you guys know that your spine or bone marrows, they're connected to the nervous system. Okay, so these type of proteins are found largely in the nervous system. Okay, so you should stay away from getting that kind of meat. If you're just eating the normal cow like beef, usually it's not as big of a problem because these type of proteins are not going to be in large quantities in meat, but more related to the nervous system. For example, brain, spinal cord, that kind of stuff. Okay, so maybe that teaches you a little bit of something to eat. So every now and then we will have mad cow disease outbreaks. Okay, we have them, and then for the most part, what people will do is they're going to kill all those cows. Okay, they're going to kill them and then get rid of their bodies. Okay, now the problem is, if we know a population of cows that have this disease, okay, and we know that the appearance, the phenotype, right, the actual disease, the symptoms don't show until you have a lot of these proteins, right? So what that means is that your cow can have some of these proteins, but then they don't display the phenotype, right? But then you still have these proteins already, okay? So that's a problem because, you know, like what if you're eating a cow that looked normal, when the butchers killed it, but then actually it already has these proteins that we don't know. Okay, so this is one of the reasons why people are afraid of these type of diseases. Okay, so this is how it works. So the PRPSC can penetrate the blood-brain barrier and then actually end up in your brain. So they get absorbed by your nervous cells. So these are your PRPCs. The normal prion proteins are produced by endoplasmic reticulum inside the cell, okay, and they're transported like that. Okay, so those are the normal. These are the normal cells. Okay, the normal PRPs. The prion normal form. Okay, that's the prion SC. Okay, so the normal PRPC prions are transformed by scrapie forms to the PRPSC, and then this transformation happens again and again and again. So what you see is that the prion proteins, because it cannot be destroyed really, so it comes and make your PRPC into the scrapie protein. This is normal PRPC. You see alpha helix, beta sheet, but primarily helical structure. And then once you turn them into PRPC, PRPSC, you have more beta sheet characters. Okay? And essentially what happens then is they form aggregates. Okay? This is the PRPSC beta sheets. So they form a lot of aggregates, and more PRPSC come together, and then they're going to form this beta fibro. Okay, this amyloid fibro. That looks like that. Okay? And a lot of that forms, and then it's going to disconnect your nervous system. Okay? That's the amyloid strand. They start to form plaques in the brain tissues. Okay, so they're going to form insoluble chunks in the brain tissue, and then they're going to aggregate, and that's called amyloid plaque. So you have a lot of those coming together. That's what you get. Okay, and what does that do? Okay, amyloid plaque aggregations are neurotoxic to brain. Okay, and this disease eventually leads to dementia and ultimately death, okay, because essentially it kills all of your head. That's the cruel disease that I was talking about. I mean, maybe they don't eat people, but they definitely eat like raw animal. Right? So that's why you should always cook your meat. Right? Hopefully, you know. So now we're going to get into structure prediction. Okay? So hopefully we went through a good discussion about how proteins, form, how proteins fold. Now, of course, given the modern science and modern technology, now we have the ability to predict protein structures. Why? Why do we need to predict that? Because in order for us to know the function of a protein, or at least if this protein is bad for you, we can make drug for it, or if this protein is useful, we can engineer this protein. So in order to do that, we need to have the structure of the protein, okay? And we're going to talk about how to predict the structures. Now, protein secondary structures can now be predicted with good accuracy. Tertiary structure, however, is not as accurate, but we can get some useful approximation. How do we predict things? Okay, so this is actually a learning process for machines. So the more known structures we have, the more accurate we can design our software and models to predict protein structures. And number one is we can look at amino acid spread. Okay, so what you can see is that some amino acids, these guys, favor alpha helix, because the probability of alpha helix having these amino acids is higher than beta sheets, higher than turns. Similarly, you have these guys that favor beta sheets. These guys favor turns. So based on the amino acid sequence, you can already know that some residues likes alpha helix, some like beta sheets.